Hello everyone. Today's topic is abnormal red blood cell. I will discuss this topic in details. If you like this video, please like, comment and subscribe my channel and also share it with your friends. There is some introduction of red blood cell. First of all, what is red blood cell? Red blood cell are the cellular component of the blood involved in the transportation of gases and nutrition throughout the human body. So, red blood cell are the cellular component. Means red blood cell is a type of the cell that is present in the blood and also helps the transportation of gases like oxygen from the lung to the all tissues or carbon dioxide from all tissues to the lung, and it also helps to carry nutrition throughout our whole body. Then the shape of RBC. The normal shape of RBC is circular, non-nucleated and biconcave shape. The center is thin, minimum 0.8 micrometer in diameter, and the periphery is thick, approximately 2 micrometer. You can see in this picture that there is a RBC and the total diameter of RBC is approximately 7 to 8 micrometer. If you cut it from a middle position and take a pieces of the RBC, then it look like a dumbbell. This is a dumbbell shape RBC. After the cut of a RBC from its middle portion, we can find this dumbbell shaped RBC, and the center thickness is 0.8 micrometer and periphery is 2 micrometer. The properties of RBC. 1. Shape. The normal shape of RBC is biconcave. Then size 7.5 micrometer in diameter. Thickness 1 micrometer at center and 2 micrometer at periphery. Then volume 87 micrometer cube. Then the composition of RBC. First one is water, hemoglobin and other substrate include glucose, protein, lipid, enzyme. In the RBC there are 62.5% water, 35% hemoglobin and 2.5% other substance. Then the morphology of RBC. The morphology of RBC is normocytic, normochromic, where normo means normal and cytic means size. Means normal size of RBC. The normal size of RBC is 7.5 micrometer in diameter and the shape is biconcave shape. Then normochromic, where normo means normal and chromic means color. So the RBC's color should be red. Then the lifespan of RBC. The normal lifespan of RBC is 120 days. The normal range of RBC in case of adult it must be 4 to 6 million per cubic meter, and in case of adult it must be 4 to 5 million per cubic meter. In children 3 to 5 million per cubic meter. In case of a newborn baby 6 to 8 million per cubic meter. Then this slide, abnormal red blood cell. There are mainly 4 types of the abnormality occurs in the red blood cell. Those are Anisocytosis, Poikilocytosis, variation in color and inclusion in RBC. A. Anisocytosis. When variation occurs in the size of the RBC is called anisocytosis. Then B. Poikilocytosis. When variation occurs in the shape of the RBC is called poikilocytosis. Then the variation in color that can be divided into 2 types, hypochromic and hyperchromic. Hypochromic, where the color of the RBC become decreased, and in case of the hyperchromic, where the color of the RBC become increased. And then the D. Inclusion in RBC, abnormal structure present in the RBC which having certain staining characteristic. The causes of the presence of abnormal RBC. There are some common causes of the abnormal RBC. First one, abnormal hemoglobin formation, abnormal erythropoiesis, RBC damage after production, decreased hemoglobin production, and autoimmune disease. Anisocytosis. So what is anisocytosis? Anisocytosis is a variation in the red blood cell diameter on a blood film, where aniso means unequal and cytosis means a cell. In this case, red blood cell. A heterogenic population of erythrocyte. Red blood cell distribution width is also called RDW is greater than 14.5%. In anisocytosis, then it can be divided into 3 types: normocytosis, macrocytosis and microcytosis. In this picture you can see the size variation of RBC. microcyte, normocyte and macrocyte. In the microcyte, the size of the RBC is smaller than the normal. In the normocyte, the size of the RBC is normal, and in the macrocyte, the size of the RBC is greater than the normal size of RBC. Normocytosis. When the size of the RBC is normal is called normocytosis. The volume of red blood cell is about 78 micrometer cube and the diameter is 7.5 micrometer. It is a smooth, non-nucleated biconcave dish, and the mean corpuscular volume or the mean cell volume MCV of the normocyte is 80 to 100 femtoliter. Second types of the anisocytosis is microcytosis. When the average size of the red blood cell is less than normal in the blood film is termed microcytosis. The volume of the red blood cell is less than 80 micrometer cube and its mean cell volume or MCV is less than 80 femtoliter. And in the microcytosis, the red cell smaller than 7 micrometer in diameter. Then the causes of the microcytosis: iron deficiency anemia, thalassemia, lead poisoning, sideroblastic anemia and the anemia of the chronic disease. Those are the causes of the microcytosis. Then the last types of the anisocytosis is macrocytosis. When the average size of the red blood cell are more than the normal size of the red blood cell in the blood film is termed as macrocytosis. In the macrocytosis, the red blood cell are longer than 9 micrometer in diameter. Then RBCs are the round or oval shape. Mean cell volume or MCV is greater than 500 femtoliter, and in the macrocytosis the volume of the red blood cell are more than 94 micrometer cube. Then the causes of the macrocytosis. There are the lot of causes. Those are in megaloblastic anemia, aplastic anemia, folate deficiency, liver, reticulocytosis, obstruction airway disease and excess of the alcohol consumption can be causes the formation of macrocyte. The second type of abnormality of the red blood cell is poikilocytosis. So what is poikilocytosis? Poikilocytosis is an abnormal shape of RBC in the blood. More than 10% of the red blood cell in a blood sample are normal in shape. Poikilocytes can be flat, elongated, sickle shape or irregular contracted. It is usually seen when older cells are present in the blood circulation. Types of poikilocytosis. These all are the types of poikilocytosis. Those are spherocytosis, ovalpcytes, burr cell or acanthocyte, teardrop cell, acanthocytes, steatocytes, sickle cell, target cell, pencil cell, elliptocytes, blister cell, keratocytes, stomatocyte, fragment and rouleux formation. These all are discussed from the next slide one by one. The first types of the poikilocytosis is spherocytosis. In the spherocytosis, the red blood cell or spherocyte are more spherical and small cell. And in the spherocyte, there is a lack of central area of the polar on a stained blood film. You see this in this picture that this is a picture of spherocytes. There is no central area and all the cell are stained by the deep stain. Later, what are the causes of spherocytosis? It can be hemolytic anemia, hereditary spherocytosis, snake venom, infection with C. perfringens is also called Clostridium perfringens, drug induces. Those are all of the problem which can create spherocyte cell. The second types of poikilocytes are ovalpcytes. The shape of the RBC become oval. In a normal blood film, about 10% of the RBC are ovalpcytes. The tail end of the red blood cell appear oval shape and elliptical shape also found commonly in the red blood cell. Then what are the causes of ovalpcyte? There are mainly four causes. Those are thalassemia major, iron deficiency anemia, sickle cell anemia and hereditary ovalpcytes. In all of these problems, the red blood cell shaped become oval. The third type is burr cell or echinocyte, where the red blood cell is uniformly spread and pointed projection present on their surface. The causes of the burr-shaped red blood cell is uremia, where abnormally high level of the waste product present in the blood, and it may be urea creatinine. Then the second point is acute blood loss, hemolytic anemia. Those are the some causes where our red cell become burr-shaped. You can see in this picture that the membrane of the RBC are not plain. They form a burr-shaped structure. The another types of the poikilocytosis is teardrop cells, where the teardrops or pear-shaped RBC present in the red blood cell. Causes of the teardrop cells: their iron deficiency, bone marrow fibrosis, thalassemia, where red blood cell form are teardrop shaped. Acanthocyte, where irregular craniated cells are found with the irregular spread shaped projection. The causes of the acanthocyte form of the red blood cell are liver disease, abnormal phospholipid membrane, inherited abnormality of the red blood cell membrane protein, post-splenectomy. In all of these disorders, acanthocyte red blood cells are formed. Another types of the poikilocytosis is histocytes, where the fragmented red blood cells are present on the various size and shaped. The causes are uremia, red cell damage due to the burn or any drugs, macroangiopathy, hemolytic anemia. In all of these disorders, where our red blood cell become fragmented, and we found fragmented red blood cell in the blood film. Another types of the poikilocytosis is sickle cell, where the sickle shaped red blood cells are found, which is elongated and deeply stained red blood cell with a pointed end. The causes of the sickle cells are sickle cell anemia, it also called HbS disease or hemoglobin S disease, because hemoglobin S, a types of the hemoglobin which is present in the sickle cell anemia, and another causes is thalassemia. Target cells. These are also types of the poikilocytosis, where our red blood cell become hypochromic, where the color of the central area of the red blood cell become decrease. So in the central of the hypochromic RBCs, there is a small pots appear, which is made up of hemoglobin and form a target board like structure. That's why it is called target cell. So what are the causes of the formation of target cell? First of all, thalassemia, then hemoglobinopathies, where hemoglobin S and hemoglobin C are formed, which is the abnormal types of the hemoglobin, and due to the presence of these target cells are formed. Then sickle cell anemia, liver disease, post-splenectomy, those are also causes due to the formation of target cell. Pencil cell, where elongated narrow red blood cells are found in our blood film. It is shaped like a pencil, so it's called pencil cells, and it is usually found in iron deficiency anemia. The another types is elliptocytes, which is the oval shaped red blood cell, where long axis is twist the short axis. The causes of the elliptocytes are hereditary elliptocytosis, thalassemia, megaloblastic anemia, iron deficiency anemia. Those are the causes of the formation of elliptocytes. Blister cell. It is a type of the red blood cell where eccentric hollow area are present in the red blood cell. You can see in this picture that there is the red blood cell and a hollow area that is present on the periphery of the red blood cell, not in the central area of the red blood cell. Blister cells are usually found in the microangiopathy, hemolytic anemia. Then keratocytes or horn cell. It is also called a bite cell or helmet cells. In this case, the red blood cell has a pair of spicules. It is a half moon or helmet with a blister-like projection or two horns-like projection. It's also called hamlet cell or bite cell, because in the red blood cell there is present a bite-like structure. That's why it's also called bite cells, but it remains in the circulation only few hours. Then the causes of the horn cells or keratocyte cells. The causes of the formation of keratocytes are mechanical damage, removal of hinge body by pleating action of the spleen, uremia, or due to the any EDTA artifacts. Stomatocyte, which is another abnormal shape of the RBC, where mouth-like shape of the RBC are formed. In a dried film, RBC having a slit or stoma-like areas at the central of the cells, and in the wet film, cells appears like a cup. Then the next point is, what are the causes of the formation of stomatocyte cell? In the hemolytic anemia, liver disease or alcoholism, the stomatocyte cells are formed. The last point of the poikilocytosis is rouleux formation, where the RBC are arranged in a row like a stick of coins. You can see in this picture that the RBC form a rouleux form. It is also like a sticks of coins. In the manual method of the ESR or erythrocyte sedimentation rate test, you see the rouleux formation of RBC in the Westergren tube. In acute or chronic inflammatory disorder or plasma cell myeloma, we found rouleux form of RBC. So I think this video is getting too long, so I end this session here. The next part of this video I will discuss in next another video. If this video is helpful to you, please like, comment and subscribe my channel if you want the next part as soon as possible, and also share it with your friends. Thank you everyone.